Okay, welcome everybody. It's wonderful to see everybody in person. It's been a long time since we've had the chance to actually see each other in person, and it's my pleasure to introduce Peter, who is from the episodic memory group, and he's going to be giving us a primer on BID's format for fMRI data and automated pre-processing with fMRiprep. So over to you, Peter.
Hello, everyone. So just a quick overview of what I'll be talking about today. So I'll just talk briefly about what BIDS is and like the fMRiprep and the main advantages of using it. And then I'll go into a bit more detail of basically how to convert from the DICOMs to BIDS format and then how to do the fMRiprep. So basically, the first bit is the general overview, where if people don't like it, they can leave afterwards if they're not interested how it actually works. So let's go into it. So what's the point of these types of project is to increase reproducibility and data sharing. So the main point of having like a standard data format for whatever data you're using is that then it is simply easier to share your data with somebody else. If there's a new student coming or internationally, or even if you just are rerunning analysis on a project that you have collected the data 5 years ago, it's much easier to remember what was happening if you have like exactly the same data structure. So the main idea of BIDS is exactly this, to standardize the same format for different types of data. So BIDS is not just for fMRI data. It has extensions also for EEG, MEG, ECOG. So that's like implanted electrodes for epilepsy patients. And there's like even some plans for expansions for like behavioral data or computational models or things like that. However, I've mostly worked with fMRI data, so I'll be talking about BIDS for fMRI data. It's mainly been developed by Russ Poldrack, and Chris Gorgulewski is the first author of the paper. And throughout the slides, I basically have some useful links where people can click on afterwards and see like a bit more details about these things. So BIDS format is BIDS format is probably quite similar to how most people store their data already, I believe. At least it was quite similar for the way I store my data. But effectively, the idea is that from a DICOM format, you can transfer into a format that is basically human and machine readable and understandable. So you have like a folder, which is like your project or data set. You have like Excel sheets with participants' demographics. You have like for each subject a separate folder. And then within that subject, you have the different data types. So anatomical, functional, field maps, if you have field maps, if you have tractography, you can have tractography. Basically, each different modality is a separate folder, which also makes it useful if you have like fMRI and EEG combination of something, or fMRI and tractography. In terms of as example, this is pretty much how like an example data set might look like. So this is like taken from Open Neuro. So the DS is the project folder. Then you just have the dataset description. So if you're sharing, somebody can literally open that file. It's like a text file. You can read it and see some metadata about the project. Effectively, something like this is a episodic memory project with task 1 created by Petar Raykov. Participant demographics, and anatomy, functional images, and so on. You often have like the name of the task as well. So it's a pretty standard. If you have different tasks, then they all just go in the same folder, and having different runs, they all go in the same folder. So one thing that is basically specific about the bits dataset is that you get JSON files like this that contain information, meta information from the DICOMs that are not really stored in the NIfTY. So that would be like the recording parameters, the sequence parameters, effectively. And these sequence parameters are used by BIDS apps. So to effectively run automatic pre-processing, so to speak. You can see how automatic it is in a bit, but the main difference between just running a DICOM to NII and then storing these NIfTY files that we often use for fMRI is that you also have these JSON files that contain the sequence parameters and whatnot. So there's very different ways how to, so as you can probably notice, I'll soon switch to the practical stuff, but like there's many different ways that you can convert the DICOMs to NIfTY format. So there's a lot of software around and they all kind of do the same thing. One very straightforward way would be to just use DICOM to NIX. There is an option where you just click save the JSON file and then do the restructuring into the folders manually or with a script that you write yourself. These other options try to simplify that slightly by you create the structure, they know the structure of the bits format, so you don't have to specify that. So you just have to say, I have a functional, anatomical, and field map images, and then they know the bits format, so they just convert the DICOMs because they can read the information from the DICOMs and say, okay, this is an anatomical image, this is a functional image, and whatnot. So I'll be talking about HUDICOP, which I'm going to show you how to do that in practice. There is another option for converting DICOMs directly to BIDS, but this is like it's having to do with like reprogramming something in the Siemens scanner so that it makes it even easier to convert the DICOMs to BID. Because for the first way, effectively for each separate project, it does require a bit of manual intervention of like writing like 10 lines of code, whereas for these bits with repro in, if that was set up, it's even easier because then it doesn't at all require manual intervention. Basically, you get bits format directly from the scanner. So a brief side note here is about Docker containers. So this is for all extensive purposes like a virtual machine. And they're quite useful because you can run software from other people without caring about the installation and these things. You're basically just downloading as if you're downloading the exact versions of software that they're using. And that is quite useful for reproducibility and making sure that things work in exactly the manner that they're supposed to work. Indeed, Chris Racey has set up Docker for the analysis server. So that's why I'm mentioning it, because it is used throughout. So for now, I'll basically go to the practical bit of going to the analysis server. So Chris Racey has shared a folder in the SISC2 volume shared, and it's in fMRiprep demo. So hopefully everybody on that should be able to get to that folder, and whenever they want to convert their bits format, see the structure files and code and everything. So the fMRiprep demo is the dataset folder. So this is your main project folder. And whenever you're doing like checks that, for example, your data is in bits format, it's this folder is the thing that you put in. And there's like basically software. It's like even online, you could just Google bits validator and you just upload that folder online and it will tell you whether your data is in bits format or not. So the main way that the folder is organized is that you have the raw images. So you still have to copy them from the CISC Dropboxes and put them in like a raw folder. And that raw folder should be somewhat organized logically, such as you have subject 1, subject 2, subject 3. And then I have here just created some text files that like for the CISC number, if I ever need the CISC number, this is my CISC number, so that I don't lose any information from the CISC Dropbox to the bits format conversion. So basically, you have just the DICOMs in this folder that are copy pasted from the Dropbox. And the idea is that if this is your first subject, so this is when the manual intervention comes with Hudikov, is that you have to run. So Hudikov is installed on the analysis server, so you don't have to worry how to install it or anything like that. It's basically a Python script that if you're using on your own computer, you can also run with Docker. So these things are commented out, but basically, I've used Docker before to use UDConf. For now, the main program code to run is a single line of code that, to translate it, means UDConf D for directory, then you put the directory where the subject and DICOMs are. So here, this is an argument for subject, so that I don't have to go every time and put subject 1, 2, 3, 4. So it takes this argument and puts it here. But otherwise, this is just the directory of where the DICOMs are. That's why it's useful to always have subject 1, 2, 3, 4, and then session is taken from here. And so -s is the subject argument, and -ss is the session argument. And then they get, it's as if I have written this bit effectively. The output is just output directory, which is you have like a separate nifty folder where you're going to convert the bits format correctly. Which is, you just copy that directory and you just write convert all. So subject session, and CNI means that the first time you run it, it doesn't do any conversion effectively. So this is a bit confusing because it says convert all and then CNI. But like for the first subject you run the project, you initially don't run the actual conversion. You run it to create a hidden folder in which it creates a heuristic py file and a DICOM to session TSV file. So it's basically an Excel sheet. And in this Excel sheet, what UDConf has done, it has read all the DICOMs that I have in that particular subjects folder, and has separated them by the series ID and session numbers, and basically it has grouped them effectively into the different types of scans. So now having this Excel sheet is actually quite important, because the heuristic, I'm going to minimize that so I can show the same two things at once. This is okay. I'm not going to minimize it. We're going to try our working memory. So basically, in that Excel sheet, you have all the, like, quite a lot of information about the DICOMs. So you have the series files, you have like the dimensions. So the dimensions are important if, for example, the fourth dimension is time, by the way. So if somebody has started run 1 but has done only 10 runs or something, then you can effectively include that in your heuristic file so that you ignore that one because maybe you don't care about 10 volumes of scanning data. And effectively, the important bit is the series ID, because that tells you these DICOM numbers, which are what you get from CISC, are related to this type of image. And with this series ID, you can use the heuristic pi script that I will just show you now to do the conversion automatically. And the idea is that you would probably have the same series ID for every subject, and that's why this type of scripting works. But these files, by the way, probably shouldn't necessarily be shared across too many people, because they might have like some identifying information such as age, sex, the person that ran the project, physician. Well, the physician is not here, but you can see that it could be there, and whatnot. So this, based on this Excel sheet and this empty heuristic file. So the heuristic file is created by Hudikov and now is empty. So what this script does is literally it reads the Excel sheet, and then you have to write the code. Okay, I want these bits are my functional data, these bits is my anatomical data, and whatnot. So after you run Hudikov the first time, it creates these two files, plus other things, but they're not really important. So what you often do is you copy these files in your code directory, and you complete the information that you need, such as effectively you create like this Python script that has the bits structure, such as you have the subject in anatomical and subject anatomical T1 W. So effectively you have to write the script for how to convert the bits conversion for the first subject, and then you can run it for the other subjects afterwards. So that's where I meant that there is some manual intervention for the first subject. So basically I have like, for this subject, which is me, I have the anatomical file, functional files, which are spin echo files. That's why you have the two directions. And I have the one single task, which is the surprise task, and the reference image that comes with the sequence, which is just like a higher contrast image. So effectively, these things documented are what Hudikov created by itself, and these things here are what I wrote myself. So you write out the bits format, so you can see that it goes to the subject anatomical subject field map folder, and then the correct name for the file, so that it does follow the bits formatting. And so these things would be empty keys right now. And in this loop, sequence info is the TSV file. It just reads row by row each of the Excel rows, and then you just literally check, okay, in protocol name, I want the anatomical to be with this name, with this dimension, and like in the series description to have something. So you basically just create conditionals so that here is the protocol name. This is the, this column is the protocol name. And because it's exactly the same with our sequence, we get two anatomical files. So just based on the anatomical name, the protocol name, I can't differentiate between the two anatomical files. So that's why I include something else here that differentiates them, such as the series description. So all you have to do is literally create these conditional files to select the nifties that you want. Yeah, Sam?
Did you create the Python's function called create key? Is that yours?
No. I think that is created automatically. Yeah, do you see?
Yeah.
So you don't have to worry about that. All you have to do is basically specify the bit format for the file that you want. And then create the conversion conditionals. So here, for the tasks one, for example, I've set not only the protocol name, but also if it only selects runs that have more than 10 volumes. So if somebody started and stopped, then it would ignore that. So the main manual intervention basically is creating this. And whether you use Udikov or you write your own script, effectively, it's the same procedure. Like if you have just a nifty file somewhere and you want to structure them into bits, it's exactly the same. You're just saying, okay, these nifty files are my functional maps, so they go into the fmap, the func folder, and then whatnot. However, the slightly easier thing here is that you create it for one project once, and then you simply run that script for every next subject. So this is where the second step comes, where you run Udikov again, pretty much with exactly the same arguments, apart from now you tell it to use your new script that you have written for that particular project, and then to use dichotomy, and these things are not really important. Basically, after you create this Python script, you tell Udikov how to organize your data into bits format, you run it, and for each subject, then it would automatically go to the raw folder, the dicoms, and convert them into like a nifty structured data set. So it would create all these things. This is like it reads automatically my age and sex and whatever. If you want something such as like note, you can always just do that. These JSON files are basically mostly for sharing or after the project is finished, that you can complete them. But if you want, you can just say like, and whatever. These are just like metadata for the project. So if you're sharing it with somebody, they know who created it, and like potentially if you're worried about authorship or somebody stealing data. And it then creates automatically also these subject folders, which have the anatomical files. As I said, the main difference between bits and normal files is that so everything is zipped, so it does use up a bit less space. However, if you're using SPM, you would probably need to unzip them because SPM for some reason doesn't read zip files. And the other difference is these JSON files that are literally the acquisition parameters for that particular sequence. So if, for example, you want something, you can always just open that file and see the parameters. The problem is that if you're sharing again, potentially you might want to remove that it was from Python or something, but that's up to you. And that's pretty much the bits file conversion. So the main thing is that for the first subject, you kind of have to write that because we don't have the reprin set up. If that was set up, you wouldn't have to do that. The main point of these JSON files is that now a script can go and read these parameter files and use them for analysis. So I'm going to basically show you already run script, but first I'll just slightly talk about fMRI prep. So fMRI prep is one of these BIDS apps that uses these JSON files to read information about your project, about the sequences, and apply pre-processing that is based on heuristics and like standard rules throughout the field of what is the best way to pre-process the data. So obviously, because it's automatic, it might not necessarily be always the best, but in my experience with healthy subjects, it tends to be quite good. So it was again developed by Esteban, who works with Raspodrak and Chris Korgolewski. And it is pretty much a script that, like, for example, a postdoc or Chris Racey might have written for a particular project, where it runs through the motion correction, normalization, field map distortion, and converting everything to MNI space. And the main idea or benefit of it is that it's relatively automatic, such as that you don't have to, for each next project, write a new script, but because the information is all in the JSON files from the bits format, you literally need a single line of code, which is like fMRI prep subject 1, pretty much, to run the pre-processing for that subject. And in terms of like the options for the pre-processing, they're preset. So that to some extent prevents some like problems with repeating the same analysis again, or like somebody fiddling up with the data or deciding on certain issues. So I think like for people that have used FSL, one thing that often is problematic is that the bet, sorry, for the people that have, this is like a brain extraction step where it just removes the skull from the fMRI image. And that step often, I feel, requires a bit of manual intervention where your own bet, it doesn't really remove the whole skull, or like it removes too much. And then you have to just start playing around with the parameters and spend hours and hours on that. But basically using procedures like fMRI prep can slightly reduce that and reduce the amount of time of writing new code for each individual project. Everybody's using the same thing, so that also makes it slightly easier. Like if a new student comes or something, I don't have to care that they have messed up the field map distortion correction because they wrote something and like looked something on a website and messed it up. Like I know that they have to care just about the main analysis bit and not so much about the pre-processing. In terms of the, like, basically, it's like a script, a very detailed script that it's not impossible for somebody to write that for your project. Definitely, it's nothing super fancy, but it's written to work with multiple different projects, making it slightly easier to reproduce and share. And like if somebody has run that pre-processing, now I know exactly where the data is, and I can do, I know what is happening basically, and do whatever I want on that data. So in terms of like the documentation, it's quite well documented, explaining all the steps, and they're chosen by the developers, such as like to not use BET, which is probably a very good thing, and to use the ANT brain extraction. And they have developed, they have slightly tweaked some of these things to work better across different data sets, such as the brain extraction thing. I haven't had an issue of that it doesn't extract the brain correctly. And basically, it does explain all the options here that are used to run the pre-processing. And they're often based on what would work best with some of the other pipelines, some of the other steps in the pipeline. And pretty much I'm going to, yeah.
Specific versions of the software, so like if you took a cell or freezer and get quite different.
Yeah, so just like the different versions, there are unfortunately different versions of fMRI prep. So they're continuously improving it, but like they're at least doing a bit of like version checks themselves. So in a sense, part of the job of Esteban and these people is like bug fixing and like with each version to do something. It's definitely not the case that it's completely fail safe, such as that, I don't know, like the fMRI prep from 5 years ago. If there was fMRI prep 5 years ago, it would not have a bug. Yes. Yes, yeah, exactly. They're there, and then the idea is to always put the version number rather than latest, because if you put the latest, let's say that in the middle of my project, there was COVID, especially. So there was 2 years in the middle of my project. Now there's a new fMRI prep version, and half my participants are reprocessed with a slightly different version, which might not make absolutely any difference, but it might make a difference. So you can always download with Docker different versions. And for the people that forgot what Docker was, that's like a virtual machine. And the idea is that they have created all this software into a virtual machine. You download the virtual machine and then you run it. And you only need to install one thing, which is the Docker container. So you don't have to care about installing ANTs, FSL, AFNI, and all these things, and caring about the versions of Python and whatever, which if you've ever tried doing, it's quite nice. Yeah, I'm going to show you the basic outputs now of fMRI prep. So after you've run fMRI prep, it creates this derivative folders. So you always separate the raw Nifty files from the preprocessed data, which is easy if you want to run different types of preprocessing. The MRI-QC is something that I would not talk about today, but it is something that you can run before fMRI prep to see basically whether it makes any sense to run fMRI prep. It's like a quality check for the MRI images. So if like somebody has really dirty data, you can just skip the fMRI prep and not waste your time analyzing them. You have the fMRI prep folder where I have only one subject here, but if you have more subjects, they're all here in separate folders. What you get within this folder is, again, everything separated into anatomical files. So it's all explained in the documentation, but for the anatomical files, you get like the anatomical files into MNI, the gray matter, white matter segmentations, the cleaned up anatomical files, and whatnot. If you have run FreeSurfer as well, you can have like a FreeSurfer folder where you have the anatomical files pre-processed with FreeSurfer, which basically in fMRI prep is an option. So you can always disable or enable running FreeSurfer. For the functional files, we just have the files in MNI space that are pre-processed. You get also some confound variables, which I'll just show you in a bit in the main output. I have run ICA aroma here. So that is like an automatic denoising method where it runs independent component analysis on both images. Then it has automatic classifiers to distinguish which components are noise and which are not noise. So that again is an option. So if you don't want to run that, you can just not run it. The default option is not to run it, by the way.
That's a question about that. in other ICA denoising steps, there's often a manual intervention process where you have to kind of assess whether each of the components are signal or not. Has that been standardized for this, where you don't have to do that, it just does it all automatically? And then before you answer, did the people at home hear that question? Yes, they did.
Okay, so basically the ICA is done by FSL Melodic, and then you can do the manual intervention if you want. The ICA aroma step is literally automating it, such as that. So you run the ICA in a MNI space, and it uses like certain features, such as like how correlated is the component time series with the motion parameters, for example, or like they have some spatial features like, okay, if it's a cardiac noise, it tends to be like activation around the pons and negative activation around the frontal lobe or something like that. So it has these automatic features that it tries to automatically say this is a noise component and this is a correct component. However, it is the case that you might not like what it tells you, or it might make a slight mistake, because these are methods that were, I mean, I think the paper for ICA Roma was in 2015, and automatic methods are not necessarily always going to be perfect. So you either say, okay, I'm just going to be fine with the ICA Roma thing and do no manual intervention, or you just do the manual independent component denoising. The good thing is that it does produce like a map showing you its decision. It also has like the these Excel sheets where it tells you the component time courses for the noise components, for the correct components. It gives you the like pre-processed, if you regress out the noise components, which is this file here. So that's the non-aggressive denoising. And like if you run some of these things, basically it gives you a few options. So let's say that I didn't like the ICA aroma thing for this subject, then I can just ignore that file and go with the main file, which is just the normally pre-processed data, because the ICA aroma is run after the data is normalized. So effectively, if I just choose this file, it is as if ICA aroma was not run. If I choose this file, which is smoothed with 6 millimeters, because that's what ICA aroma does, and regressed out the noise components, then I agree with ICA aroma. Or given that I have everything in an Excel sheet, I can then manually say, okay, these components are signal, these are noise, and do only the regression of the noise components rather than the whole pre-processing procedure. And so apart from these files, what is basically quite nice about fMRiprep is that for each subject, you get an HTML file with visual outputs. So you have like these files that are, sorry, maybe you can't really read what it says here, but there is summary, such as subject 1 was run, anatomical functions, this and this was run.
Let's just ask a question. What's the difference between aggressive and non-aggressive denoising?
So the difference between aggressive and non-aggressive was that it's just how you run the regression, whether you include only the noise components and then you regress out any shared variance between the noise components, and if the noise component is correlated with the non-noise component, then that shared variance would be attributed to the noise component, which would be the aggressive one. Then the non-aggressive one is you have the noise component, but you also have the signal component in your regression, so that the shared variance is not taken up by the noise components, supposedly. And like you effectively, it's a bit like partialing out only the thing that you're most sure is noise, is the non-aggressive one. Whereas the aggressive one, anything that remotely looks like the noise, remove it. And the default is to use the non-aggressive, but you can just put an option and it would run the aggressive one. if you don't want to run the regression yourself as well. Like this is again just an option you can put in fMRIprep. And so the outputs of fMRIprep are in these HTML files. It's quite useful because it provides visual information of whether it worked or not. You probably can't see the text. Okay, I think that slightly fixed it. But if you forget what the red and blue are, it kind of tells you, although here it's a bit obvious, like the red is the gray matter and the blue is the, so the red is the skull and the blue is the boundary between gray and white matter. So you have like some visual inspection. You can see that here it didn't work perfectly, but you can decide based on preferences. So one thing to notice about my weird brain is that the fMRIprep doesn't necessarily have a completely automatic way of deciding whether to remove a subject or not. So it's the people that have created that have suggested you do some semi-visual inspection of the data, just partly because often they think that a lot of the automatic ways may not necessarily be super reproducible and reliable. And that's why it creates such nice visual reports of the data to examine, and you can agree with the pre-processing or not. The problem is if you don't agree, is that then you probably have to rerun fMRIprep again, and you might have to fiddle with some of the options. So although it's automatic, as you can probably have already noticed, there are some options to choose between, like such as you can say ignore the field map correction, or don't run slice timing correction, or don't run FreeSurf or ICAroma. So there is some flexibility still, mainly because quite often different data sets have different needs. So they've tried to maintain some flexibility, which I guess is slightly different from the human connectome preprocessing pipelines, or like some of the other consortium, like the Childman Institute preprocessing pipelines, where they have kind of assumed that you have collected exactly the same data set as them, using like the workbench and exactly the same thing. Whereas here, there is a bit more flexibility of assuming what was collected and when was collected. So in that sense, comparing it to other such pipelines, this is slightly more flexible to different types of data sets. And basically, it does have quite nice visual features to double check whether the normalization has worked. One thing that is quite neat is, so you have, that's the co-registration step with the field map distortion. So you can see whether that's worked and basically make a visual inspection. So it has for each step like a visual check where you can see whether it works or not. So this is like the co-registration between anatomical and functional. It's one thing that is neat is that it runs automatically. It creates these confound files that are all stored into an Excel sheet, which is the motion parameters. So you have the 6 motion parameters, but you also have like their derivatives and their squares. So if you want to use the 24 motion parameters, you have that. It also runs like this ComCore analysis, which is basically it gets the white matter, and so the one is the anatomical ComCore, which is literally a mask of the white matter and CSF, and running like the time series from these white matter ROIs are regressed out effectively. I mean, you do a PCA on these time series, get noise components that you can then regress out to have like slightly cleaner data set. But this is not done, right? This is all stored into an Excel sheet where you have like about 200 noise components, and it's up to you choose what you want to regress. So there is again some flexibility. If you do like functional connectivity, you have the global signal. So if you want to do a global signal regression, in terms of that, that's quite easy, because it's not the case that some pre-processing pipelines, you run it, you have the normalized images, but then now you actually you don't have the frame-wise displacement, and now you have to do the motion correction specifically to get the frame-wise displacements or something like that. Basically, it does store quite a lot of these information in an Excel sheet, so you can use it if you want to. It does create these, like, again, briefly for people that don't know what that is. So that's the frame-wise displacement. So that's like the derivative of the motion parameters. So that detects quick motion. Basically, if somebody was like made a quick jerk between TR to TR, the DVARS is pretty much the same thing. You have the global signal in the CSF, so in cerebrospinal fluid, global signal in White Panther, global signal in throughout the whole brain. These plots are quite useful. I quite like them. That's pretty much the whole brain throughout time, plotted as a matrix. And the blue thing is, for example, I think the CSF and skull. It does say it here. Like basically, whenever you forget, as me, it says like, okay, so cortical is, that's cortical boxes is blue, subcortical orange, and cerebellum is green, and white matter is red. So basically, that's quite useful because sometimes you can detect artifacts into these plots that are not seen into the time series data. So basically that should look like white noise, like a broken TV. It doesn't look like a broken TV, or there is some structure in it. It suggests that there is an artifact. And the need to think about this is that sometimes you're not going to have like frame-wise displacements that goes above a certain threshold that you would deem as an artifact and automatically remove that volume. But when you just look at that plot, you just see that there is massive structure in the data, and that probably suggests that subject is for removal. So in that sense, these plots, I find them quite useful. And you get like the correlation among noise regressors, which if you're doing like noise regression, you can see what to include in your regression. And if you run ICI-ROMA, you get these nice maps where the red means it was considered a noise component, green means it was considered a correct component. So you have the spatial map, the variance explained, the temporal frequency, and the time course. And there's a lot of components, as you can see. It does basically the same thing for every run. And a neat thing about it is that if you don't like writing, is at the end of this a very long document, you literally get the method section for the functional data preprocessing and ready. Like you can just copy paste that in your paper. So
It's actually mentioned, I think it's probably recently in here, which is absolutely crucial. Maybe no one will tell us, but just ask.
no, I mean, that's pretty much it.
There is something like that. So yeah, I think we should probably stop. I'm sorry I couldn't book a room with any space afterwards. But, is there any very, very quick questions from the people who are here?
Once you've got your pre-process fMRI data, can you plug that straight into second whatever you want, SPM or Excel?
Yeah. It's you just get the MNI normalized data. It's not even smoothed, so you might like if you want to smooth your data differently, you can do that. But yeah, you can plug it anywhere.
You mentioned about Arianna applied 6mm smoothing to the images. Is there a way to change that? Or?
No, I mean, this is just the Arianna step. That basically their features are based on the 6mm smoothing. So even if you manage to hack that, then the features won't work. So unless you write your own Arianna script, no.
Thank you very much, Peter.
Thank you.
I'll just mention there won't actually be a CNI next week. The person has had to reschedule the speaker, so it will be 2 weeks' time with Alexa is giving a talk. It's in the same room, Chris.